particles, so many tiny particles are here. If you look at the scale at the width of this particular particle, that is under 10 nm, right? So what next is that how to interpret this particular image. And after looking at the image, we may think that no, this is not a good formulation. There are degree of agglomeration over the particles. I mean, the distribution is not clearly seen. But this is not the case. Why? Because particle size is very less in this case, right? So what we can interpret that this image is a good image, is giving the uniformity, and it is showing the importance of the method as well. As I told you that these liposomes are prepared using the supercritical gas fluid method. So the pressure will be very high in that case. So it is showing the importance of depressurization stage lies in the conversion of LUVs to SUVs, means large unilamellar vesicles to small unilamellar vesicles. They are not large vesicles, they are small vesicles. So it is recommended that the depressurization to atmospheric pressure occurs at a certain temperature above the lipid phase transition to obtain the highest number of small unilamellar liposomes. Right. And what it is confirming? It is confirming that the occurrence of the particles, we are able to see hundreds of particles in this image, number one. And what it supports? It supports the formation of liposomes, and the proposed configuration for this particular method, right? And this was prepared with pressure near about 180 bar and temperature at 50 degrees centigrade. Why we cannot increase the temperature? Why? Because we are playing with the lipids in case of liposomes, so every time it must be somewhat above the phase transition temperature of the particular lipids that we have used in the formulation. Next is this particular photograph. So this is the same paper from which I took these two images. This is made using the supercritical gas fluid method, and these liposomes are made using the normal method that we use in the general practice, that is the thin film hydration method. But if you could see here, this red circle, it is showing that the film is not that much strong. I mean, the mechanical strength is poor in case of thin film hydration method, right? And the size is also bigger as compared to this particular image, right? So here the size is approximately 70 nanometer on average, and here the size was above 170 nanometer on an average, right? So we can prove our method as well. Suppose if we are preparing the liposomes via 2 or 3 methods, then which method is important? Which method is, the best method? We can interpret these things via interpreting these microscopic images. Right. So moving ahead. Now, besides the 10 photograph, these are the, besides the same photograph, these are the 10 photomicrographs of the niosomes composed of phosphatidylcholine and as a major lipid, but the authors have also used so many lipids such as the dioleal SN glycero 3 phosphocholine, that is DOPC, DPPC, DSPC, that is diacetoesteryl SN glycero phosphatidylcholine as well, right? So this is a typical picture of the niosomes. But what is the difference between these two pictures? If you look at this picture and this picture, they are identical. They are identical. Why? Because if you see, then nothing wrong in these pictures. Number one, they are identical to each other. The particle shape, particle size is quite similar. But in actual, this photograph was taken before the freeze drying, and this photograph was taken after the freeze drying. As we all know that freeze drying is very important to preserve our formulation, and sometimes it is very important. Right. So what we can interpret, what the authors have interpreted in this particular picture, that they have used these 10 photographs to visualize the effects of the freeze drying on the vesicles, lysosomal vesicles. They interpret what is happening after the lyophilization or freeze drying to the lysosomal vesicles, and they interpret that there is a no significant change after the freeze drying, right? So if we have freeze drying in our protocols, suppose we are preparing the myosomes or liposomes or any kind of nanostructure, and then we need to lyophilize them, right, or freeze drying them, then is this freeze drying is going to affect our structure in in any way, such as the morphology or topography? If it is so, then these protocols are not validated. We have to move towards the another protocol for freeze drying. Right? Why? Because some samples are compatible with drying, but some samples are not compatible with the drying. Right. So if you see in these images, right, so we can say that they are identical in nature and there is no change before and after the freeze drying. Right. Just if you look at these circles, which are due to the incorporation of the cryoprotectant, that was trehalose in that case. Now, what is this? What this figure is telling us? If you look at this particular figure, right? So this is also a TEM image of the same liposomes. Same liposomes. These are the TEM images, right? But what is the difference? And why this image is different from these kind of images? They are perfect, we can say, but this is not perfect. If you see the bilayer structure, basically by definition liposomes are bilayer structures, right? So if you see the bilayer, it is not perfect. If you see the internal structure, it's not going to tell you. And look at here, look at the arrows, what they are trying to convey. If you look at this arrow, these dark particles, here also it is the dark particle, here also it is the dark particle, here also it is the dark black particles. So what is, how to interpret this particular image, right? So this is nothing but the sampling problem. This is nothing but the sampling problem. Sometimes it happens that due to staining chemicals, we all know that certain kind of staining is required before the TEM and SEM analysis, and in any case, if the staining particles are get deposited on the surface of the nanostructure, in this case the nanoliposomes, then they may interfere with the correct morphology and topography of this particular image, right? As you can see, this is the artifacts due to the staining chemical deposition over the sample surface, and not over the sample surface but everywhere inside the dispersion medium as well. You look at, right? So this is nothing but the sample processing problem, right? So if we encounter this kind of problem, we may ask to the person where we did this particular investigation to re-carry out this particular investigation with a proper sampling procedure, or we may change the staining chemicals for these things. Right. Okay. Now, the next case, they are also liposomes. But what is the difference between these two pictures? Now, if you look at here, look at my cursor here, and if we compare this particular structure, look at my cursor, with this particular structure, what it is revealing? This particular structure, this particular structure, this particular structure with this particular structure, right? So we can simply interpret that this is a multilamellar liposomes and these are the unilamellar liposomes. When I say unilamellar, means a single bilayer is over, on the surface of the liposome. Single bilayer. So liposome is made up of single bilayer. If I say multilamellar means there are more than one layer, as in this case it is multilamellar, maybe 1, 2, 3, 4, 5, 6, maybe 10th of layer, just like onion structure, just like onion structure. One layer above one layer above one, another. Right. So this is how we can interpret this particular structure. And here it is. The next case, I included the gold nanoparticles here. Gold core, silica, shell nanoparticles. Now how to interpret these nanoparticles? If you see this 10 images, then it is clear, it is clearly showing the formation of the gold core particles, right? But how to know that these particles, these gold particles, which are in the core, and the silica particles, which are making the shell of this particular particle, I mean the outer region, right? So what the authors did here, they took this particular image as well. They magnified any region onto this picture. If you look at here, it is not clearly visible in this case, but the silica shell is clearly visible in this particular picture. Right. This silica shell is clearly visible in this particular picture. So this is silica shell, and these are the gold nanoparticle clusters. Authors have developed these kind of gold core silica nanoshells to get rid of certain cancers, such as the lymph node cancer. Okay. What next is, and not only gold particles, we may characterize in this way silver nanoparticles or any kind of metallic nanoparticles which could be conjugated with the other kind of particles such as the silica nanoparticles. Even incorporation of drug could be identified using these images. Now, this is the last case. If you look at the pictures, and this particular study was taken from scientific report, Nature scientific report, and it is telling the coating difference basically. These are the same photomicrograph of the Ebola viruses. Why? Because same is also extensively used in microbiological studies as well, to study the bacteria, to study the viruses, to study the fungi. Same is heavily or extensively used in this case. But we all know that the sputter coating is very much required in case of SEM. Before the characterization, I mean prior to characterization, the sample must be coated. So sputter coating is nothing but the deposition of the vapors of any conductive material such as the gold, silver, anything which is conductive. We actually require to make our sample conductive before SEM and TEM analysis. Right? So now how to interpret this particular image, right? And what the author did here, so to produce an electrically conductive surface for the better SEM analysis, authors have coated these Ebola virus particles with different uh, coating techniques. One is the sputter coating, and one is, which is relatively new for us, that is the ionic liquid coating, right? And they conclude which type of coating is best to know the particles under 1 micron scale or maybe 500 nanometer scale, right? So in this report, authors described the methods for concentrating the microbial suspension for the same observation on pre-coated filter substrate. If I'm talking about the sputter coating, it is pre-filter substrate. And they show that instead of sputter coating, instead of this particular coating, an ionic liquid, what is the name of that ionic liquid that is now highly considered for the same analysis uh, and replacing the sputter coating technique, right? Is 1-butyl 3-methyl imidazolium tetrafluoroborate, which can be diluted easily with the water and can be used for the rapidly infiltrate microbiological stem sample, forming an electron-lucent conductive surface, which prevents. Also, what is the advantage over this sputter coating is that this kind of coating, ionic liquid coating, is able to prevent the specimen or our sample charging and gives the good results with the microbial specimen. Right. Now, how to analyze this effect? Look at this cursor. These are the fine ultrastructural details, and basically they are defects. They are not Ebola virus. Here it is. Look at my cursor. This is Ebola virus. This is Ebola virus. Then what these structures are? These small dots. Look at the cursor. So these dots are nothing but the ultrastructural details that is due to the sputter coating. This is the coating effect. This is the coating effect and nothing to do with the sample. Nothing to do with the sample. This is just the coating effect. These fine ultrastructural details are not required during the sampling. And if you look at this ionic coating technique of the same, these ultrastructural details are, reduced very significantly. If you compare this structure with this structure, then these ultrastructural details are not found here, right? And this is just the comparison between the same photographs and 10 photographs of the same Ebola virus in the same sample the authors have used. This is a negative stain 10, how it looks like in 10 and how it looks like in same. One can identify the difference between same and 10 observations as well. Right. Even using the same, we can measure, look at the cursor, we can measure the particle size, the Ebola virus particle size, shape, dimensions, right? And this is nothing but how to interpret this particular thing. Look at my cursor. These are the clusters of the Ebola virus. These are the clusters of the Ebola virus. Right? That depends from sample to sample, right? And region to region on the sample. Okay. So okay. So let me stop this sharing and okay. So one last image of this AFM. Very different image from TEM and SEM sampling. Right? AFM. Why? Because AFM measures the samples in as a three-dimensional structures. If you look at this is the normal photographs just like SEM and TEM, but the options are available inside the software to convert this particular structure into three-dimensional structure just like this and just like this. So what is the beauty of this particular technique is that we can measure the height of the sample as well. We can measure the height of the sample as well. I have something to show you. Just give me 1 minute. So I hope my screen is visible with nanoscope analysis.
Yes, yes, it's visible.
Okay. So nanoscope is a software that is generally used to interpret the structures obtained using the AFM, and it is generally given in the CD or the pen drive from the respective place where we uh, perform our experiments, such as the IITs and NITs right now. If you look at the same the AFM photomicrograph, that is the atomic force microscopy. In biological samples, we may use the contact and non-contact mode, means the probe will be in contact with the sample, or maybe the probe is not directly in the contact with the samples. But generally, in biological samples, it is always a contact mode, right, to analyze the surface in a good manner. Right? Now, if the slide is visible, this height, my cursor. Are you able to see this cursor?
Yes, sir, it is visible.
Okay, okay.
Yes,
So,
it is
if you
visible.
look at this particular image, then this image is showing the doxorubicin entrapped graphene nanoparticles. Doxorubicin entrapped graphene nanoparticles. So how these particles looks like in the same. Now, what can be done using the AFM, not the same, sorry, it is using the AFM. Now, if you look at this left side panel, then this is the height structure that is you might have seen in so many papers. This is the height structure that we generally use to report. But there are other things as well that could be reported using the AFM. That is the peak force error, in phase, and quadrature structures. Right. Right. Same sample with the different images. Then what is the meaning of these all things? Right. So this is what that we used in general. If you look at the particular particles here, look at the cursor. This will give you the size as well, height as well. Right, I will show that as well. But first, I will explain this peak force error, in phase, and quadrature structures. What are the different types of details they are giving to us? So this peak force error, right? First, try to look at. Try to differentiate between these two images. Number one, this image. Number two, this image. And believe me, the same sample under investigation. But the type of things are different. And this peak force error is nothing but it can be compared to the deflection error in the contact mode and amplitude error in the tapping mode. Basically, it is nothing but the instrument error. It is nothing but the instrument error. If we talk about this in phase and quadrature structures, the different kind of images. If you compare this image and this in phase image, then these are the two different images, but with the same sample and the same reason these photographs are taken. Right? So this in phase and quadrature images we use when we require to demonstrate the different properties in a single sample. Why? Because if we see this particular image, we may able to see the different reason. One reason is these particles. Look at the cursor. One reason is these particles, and second reason is this dark area. This dark area is the second reason. So if you want to differentiate among the properties in the sample, then we can use this in phase or quadrature image, or maybe the editor or reviewer may require these kind of images, right? So as you can see in these images, a clear grain and boundaries, clear grain and boundaries. Example, I'm giving you one example. Suppose if you want to identify the magnetic and non-magnetic domains in the sample, you have prepared magnetic nanoparticles, but there are certain domains on your nanoparticles which are not magnetic in nature. Then how will you prove? How we can identify that yes, this region is giving the magnetic properties, or correspond to the magnetic properties, and this region is correspond to the non-magnetic region on the same nanoparticle. In that case, these particular in-phase and quadrature structures has the more important, the prime importance, right? Editor or reviewer may demand this kind of images, and Okay, so these images could be comparatively uh, supplied to the uh, it can be used in the paper as well, manuscript as well. Right. Okay, and important thing is, as I told you, this is a simple structure that we generally use to publish. How to measure the depth of the sample? One important feature of the AFM, besides all, is that we can measure the depth of the sample, depth of the nanoparticle as well. How? Look at the cursor. If you click here, the particle size, it will immediately give you this depth histogram. You could crop this particular area here, and you could publish this particular histogram along with your normal AFM figure. It will give you the more probability for the acceptance. Why? Because we are giving the depth histogram as well. What does it mean? It means it means we know the depth of our sample as well. It means we know the depth of our sample as well. How much is the depth on the NM scale, that is the nanometer scale, between 20 to 25. Right. And there are, you know, uh, many advanced feature in the software which are available, and many advanced things that we can analyze using the AFM. Right. So I'm stopping this screen share. So this is all from my side. Thank you so much uh, for patiently listening me, and my sincere thanks to the organizers, Dr. Dinesh Shah, Provost, UCA, Tarsaria University, and Dr. Ruchi Vyas, Principal Madam, and special thanks to Dr. Devesh Kapoor and his team. Thank you so much, and also the eminent faculties on board. And I'm open to any type of collaborations, review, research, etc. Contact me over here, and my email ID is here. Thank you one and all.